Our third project is, and I'm a little biased, but the most interesting, from the St. Clair region, is soil health as it relates to yield. So can we please help and welcome Amber Holland? It seems if you point it down, it works the best.
Seems if it point it down. Excellent. Whoops. Sorry about that. So next year, you're going to give this, right, Chad? No. Okay, so I inherited this kind of from Chad and from Adam. I helped with it last year. You would have heard Adam Hayes' presentation last year giving you some of the background. So I'm going to skip over. Right. Come on. Oh, there we go. Hopefully we just went one. So like everything else, is a 3-year project. We started it in 2018. 2018 was a bit of a challenge. We got 17 sites established, and of course we had dawn, which interfered with some of the harvest. But overall, the objective of the project is to assess how some of the new soil health tests that have been developed by A&L Labs correlates to yield and plant performance. And we've also put in a few other soil health tests or soil assessments to add to the data. And last year, I know Adam talked about a little bit more detail on the background of why A&L or how A&L has developed their package. We're going to dive into some different stuff today. Maybe. There we go. So here's a quick background on the budget and the partners. Of course, lots of analysis and in-kind from A&L. We've got cooperation from the Essex region, Lower Thames, and also St. Clair Region Conservation Authorities. They're helping to fund some of the soil analysis. Agris Cooperative has donated some in-kind work. Warner Ag, which is a local consulting company, also some in-kind. And Laura Vineyard's lab in particular have been very kind in helping with analysis. And in this year, they actually did all the shelling of the corn for me. And lots of OMAFRA work. Come on. So the plan. The cooperators across the 3 counties supply the field locations, and they manage the field like we're not even there. So as far as grower impact, there's very little other than the tons of pink flags and pink flagging tape all over the field and a sign out front. We get field boundary maps created. That's where Warner Ag comes in, and then Agris supplies the NDVI maps. And then the growers, of course, give me the background data and any historical yield maps that they have. Oh, come on. Oops. So that's pretty. Margaret, where are you? Isn't that a donation? There we go. The whole point of this, it builds off of some work that A&L did, right? So they were sampling multiple areas within one field, looking at good and bad or healthy and stressed. In this case, we're trying to move it across all 3 counties, different soil types, different cropping systems, but all in corn. And we're picking a good site and a bad site in each field. So Agris does the NDVI. The students and I will sit down with the NDVI maps and also the historical yield maps and try and identify historically good and bad or healthy and stressed areas so that we hopefully take a little bit of the seasonality out. And then we get there actually in person. Come on. When we get there actually in person to do the sampling, we also take a look at the landscape and whether it's just a drainage problem or something that is inherent to the soil. So the NDVI and the sampling is supposed to be completed between V10 and R1, so pretty tight window. And also my personal favorite time to be in corn. When the van is yellow, it's lovely. And so for each of the good and bad areas, we're taking 5 root samples, so the whole root ball, which means we're walking across full-on cornfields with buckets of root balls. We're taking soil samples, we're staging the crop of each of those 5 plants. We're also taking the depth of the A horizon and trying to get GPS coordinates. So soil sampling, also really fun carrying soil sample probes through full-on corn. Oops, there we go. The students are in there packaging up the corn balls right there. We tag each one because when it goes to the lab extracts soil from each root ball. And that's what they're doing their analysis on. So each root ball actually has the full soil health test done on it. And then select locations are also having a DNA analysis run on it. And we've got this down to a science. We no longer take watermelon-sized samples back to the lab. We now take something that's a lot easier to carry. As we realized fairly quickly, they don't need watermelon-sized samples, as much as they appreciated getting all that extra soil. Everything tagged separately, so they identify it. Come on. And we have the lovely plot cooperator signs on the outside, which helps to remind the growers that this is where their plot is, too. And then the samples are delivered to A&L within a day or two, and they're held cold until they're analyzed. Then, after the students have disappeared, then we do hand harvests. And we take 2 rows, so a 1,000th of an acre from each the good and the bad areas, and I recheck the GPS location, and I carry those bags out. And some of those fields were really fun this year, as I know all of you experienced too. There's something else walking through with bags of corn on your back with corn that's pretty much flat. Not fun. All the growers collect the yield data and hopefully send it to me, which is a reminder to a few guys, because I don't have all the combine yield data yet. And then we'll extract the actual point yields from there. And soil management data is collected for each field. A&L labs do some of the analysis. We send the whole data set back to Laura and her lab there, and they'll actually run some stats on it. So I don't have the 2019 stats yet. I just have the 2018. But I do have a little bit of data I wanted to show you really quickly. So I focused on some of the things that I could pull out fairly easily. And last year in 2018, we had some issues, right? We had dawn, so there was a couple of fields that never even got machine harvested. They were just destroyed. We had hand harvest yields. This year, we did 18 fields. I have hand yields for all 18, and I expect to have machine yields for all 18 also. So here's the hand yields, and what I did here was I looked at the good versus bad, because last year in 2018, we had some real issues in picking the good versus the bad. If there's one thing I think we did a little bit better, we found the good or the healthy versus the stressed a little bit better this year. Because you can see I have a lot more of those bars that are above rather than below. Now, I do want you to take note of 19-2, 19-6, and 19-14. Those are the 3 larger ones that are extending below the line, because we're going to take a look at a few other things. So going back to some of Laura Vanier's work looking at soil health, some of the things that she's tagged in looking at the various soil health tests that are out there as being the most representative. So this is work that was done on the long-term rotation and tillage trials of Bill Dean and Dave Hooker, so Laura and Ridgetown. Aggregate stability was tagged as one of the things that is a fairly decent predictor. So we run aggregate stabilities on each one of those soil samples that we take from all those locations. So 36 soil samples had aggregate stability run. And I know that's a whole lot of bars. The green is the good, the blue is the bad. G, B, just makes it easier for me to remember. And I've tried to kind of group them so they're a little bit easier to see. For the most part, 15 of the 18 locations had higher aggregate stability, good versus bad. Now, there's that 19-2, 19-6, and 19-14. Those are the ones that were under yielding or backwards yield. It's interesting that for the most part, it looks like their aggregate stabilities are backwards too. The other thing we looked at was the depth of A horizon. So this is looking more at some of the inherent quality of the soil. How much topsoil is really there? And we do have one wonky location where the topsoil was, I'm pretty sure it was a badly eroded spot. And again, I'm looking at differences, so good versus bad. So good being above, the line means the good was deeper in topsoil. If we have a bar that's going below, it means that the bad or stressed area had higher topsoil. And again, those are the 3 locations where we had significant yield reductions or flips. So interesting, the topsoil really wasn't telling me anything there. Solvita was one of the other things that Laura has indicated, and it's part of the Vitalis package of tests that A&L is doing as part of this project. And Solvita was one of the ones that was tagged. So again, we've got the green is the good, the blue is the bad or the stressed, and I realize it would take a lot more time to sit here and digest all that. But it's a respiration test, so it gives us an element of the biology of the soil. And there's our 3. Unfortunately, we're missing the 14s for some reason, but the 2s, there's no big difference, but in the 6, we can see that the good is better than the bad. I still need to spend more time staring at this and looking at the numbers. So going back to some of the information that came out of 2018 after Laura and her crew went through and did a much more detailed statistical analysis. They looked at the rhizosphere soil versus the bulk soil, and there were some groupings with the rhizosphere soil, and we did see higher zinc and microbial active carbon in the healthy versus the stressed, whereas the bulk soil, there's no real separation. So there's a big difference between rhizosphere soil, that stuff that's closely associated with the roots, versus the bulk soil that was taken kind of outside the root ball area. These are the kinds of analysis that they're doing for us, and the biggest part was we end up with a lack of statistical differences in yield showing up in 2018 between the healthy and the stressed sites. That was consistent with seeing very few differences in the soil health indicators. Some of that probably was we weren't doing a good job of choosing our locations, but it also was one-year data. So I'll have a whole lot more, or someone will have a whole lot more to tell you next year. Maybe it's Chad. Communications, usual presentations, newsletters, things like that, and eventually the crop advances report. Challenges, we had the same challenges everybody else did, 2019. It was wet, and then it was dry. Some of the fields where I thought we had it dead on for the good versus the bad, there was corn there that looked like it was just awesome. It was trying to put out that second cob. And then we got dry. And when I went to harvest them, some of those ones where the yields were backwards, I think it's because the stressed area didn't overstretch its ability. It only tried to make 1 cob, and some of those good areas tried to make a cob and a half and failed. So harvesting was fun. But there was some really good stuff out of this. Adam's files were so organized, it was amazing. That was awesome. Thank God for him. Cooperators were terrific. I was really concerned that these guys weren't going to let me know when they were harvesting. They were excellent. They told me, they gave me a heads up. It was great. And I had the most amazing, organized, and dedicated students. Even the one that I gave heat stroke to the one day. Whoops. So again, thanks to all our partners, and of course, Adam Hayes, our AMAFRA retiree and answer of many, many questions. Whoops. There.
So questions for Ann? All right, can we thank Ann for her presentation?